Nombreux publics, bonjour. Merci d'être présent à cette présentation des lauréats des 3 prix de la Société suisse de cardiologie, de la Fondation suisse de cardiologie et de la Société suisse de chirurgie cardiovasculaire. Si le nombre n'est pas là, la qualité des personnes dans la salle est en tout cas très élevée. Merci à vous d'être ici et je pense que c'est important de reconnaître, d'avoir cette reconnaissance par rapport à des jeunes chercheurs pour leur travail talentueux qu'on va appeler ici. Et c'est un plaisir que de co-présider cette séance avec le professeur Juni, président de la Société suisse de chirurgie cardiovasculaire, responsable du service à Zurich. C'est un plaisir et un honneur aussi pour moi d'appeler le premier lauréat qui a gagné le prix de recherche 2013 de la Fondation suisse de cardiologie, le docteur Georg Ehret de Genève, qui va nous parler de variations génétiques influençant possiblement la pression artérielle. Georg.
Dear colleagues, dear members of the Swiss Heart Foundation, it's a great pleasure to start this session of this afternoon with the first talk on these selected scientific topics. And it's a particularly great pleasure to receive this year's research prize of the Swiss Heart Foundation. Thank you very much. I'll try to summarize within 8-9 minutes a project I've been working on for a number of years now, 8-9 years. And I think the basic question is, why is blood pressure genomics relevant to our meeting this year in Lugano? I find this quote from Eric Lander very helpful, which he said in 2001, that most folks don't realize that we don't know the root cause of asthma, heart disease, diabetes, and hypertension, and this is still true today. We know covariates like body weight and others, but the true root cause why somebody in a family gets high blood pressure and why another doesn't isn't clear, but it's getting clearer over the years. And here, genetics and genomics can help. Unbiased genome-wide association studies can identify variants associated with these phenotypes, and this is the topic of this short talk. Every phenotype, and particularly also our most important cardiovascular risk factors, have a strong genetic component. As you see in the table here, that for blood pressure, this turns around 50%, for cholesterol and other phenotypes in the lipid realm, 60% or something like that, and even for coronary artery disease, around 60% heritability. We know today that most of this heritability is due to SNPs, which are single nucleotide polymorphisms, a variant in one letter of your genetic code, and every one of you has 4 million of these variants, which make most of all the phenotypes we observe today. In hypertension research, monogenic hypertensive syndromes have been particularly instructive and illuminate certain pathways which fall all and exclusively into the kidney or the steroid metabolism. And these patients are really on the extreme of the phenotypic distribution. But we are much more interested in the remainder of the distribution that you see in your clinic every day. And we know now that the monogenic hypertensive syndromes don't make up a significant proportion of this overall distribution. So what we do for genome-wide association studies is basically take a group of diseased people here, in the example in red, take a group of healthy people in blue, count genotypes in both groups, derive a statistic, chi-square statistic, and a p-value, and then do this over and over again for millions of times for all variants known in the human genome. You see immediately that this constitutes a large number of tests, and you need to adjust for the number of tests, otherwise you will get by chance alone a highly significant result. So here, the significance threshold will be typically 5 times 10 to the power minus 8, And you can imagine immediately you only get to that kind of a p-value if you have a lot of people. And that's the tricky part of these studies. Here is the earliest study of that type for blood pressure of the Wellcome Trust Case Control Consortium. They tested several phenotypes, and you can see that for CAD, for type 1 and type 2 diabetes, there were hits. So these are Manhattan plots. On the x-axis is the genomic position, and on the y-axis, the -10 log of the p-value. So the higher you get, the more significant your p-value is. And you see that for blood pressure or hypertension in this case, there wasn't any signal at all. So we now understand why this is. You need large sample sizes, and visionary people already 6-7 years ago got together all the big cohort studies from the US and also from Europe to have a very large sample size. In the end, this led to the establishment of this international consortium called International Consortium on Blood Pressure, GWAS, with a total sample size of 70,000 individuals. And our experiment, published now already more than a year ago, was somewhat complicated in design. I won't go into the details, but basically we looked at the first visit systolic and diastolic blood pressure, adjusted the phenotypes, and then meta-analyzed every single study's results across all studies. In discovery, for 3 million SNPs, we had 70,000 individuals, and there were 2 replication phases with an additional 130,000 individuals. And all this led to the discovery of 29 independent significant SNPs. Some of them were known previously in another iteration of this that we had done. If you look at the genes that are near these SNPs that are genome-wide significant, a little small for you to see, but if you go through this list, there are very few and only in bold type, genes that are previously known to be associated with blood pressure. Basically, the 2 natriuretic peptide genes, 2 cytochromes, and adrenomedulin. All the others were completely unknown to be associated with blood pressure before. Underlined and in red are genes that carry a non-synonymous coding variant, which might be causal for the blood pressure change observed. But you can see in the right panel here, where the scale goes from 0 to 1 millimeter mercury, these are the effect sizes of the 29 variants identified, that no single variant gives rise to more than 1 millimeter mercury of blood pressure. So we can conclude from this, in this very large study, that there are multiple small effect size variants that determine the overall distribution of blood pressure in the population. We then went on and looked at perform so-called Mendelian randomization experiments. We know that our 29 SNP risk score influences blood pressure, but does it also influence target organ damage on the heart, on the brain, and on the kidney? And you can see very summarily that for the heart, we get a highly significant result. So our 29 SNP risk score influences left ventricular mass and left ventricular thickness. Our 29 SNP risk score predicts CAD and stroke. It's suggestive for incident heart failure, but for the 5 kidney phenotypes, serum creatinine and others that we have tested, that there was no signal at all, which was very intriguing and might indicate that blood pressure is not driving kidney damage, but more pre-existing kidney damage, even in apparently healthy individuals, might lead to blood pressure increase. What's next? There are many things to do. It seems a little strange that we're still aiming for larger sample sizes, but given our high multiple testing burden, this is very promising. So much larger studies are ongoing. Currently, an analysis with 200,000 people is ongoing within our consortium, and there are other pushes for even higher sample sizes, particularly in the UK and other places. We also have looked only at rare SNPs for now. You see that this is an allele frequency. We stopped basically at a minor allele frequency of 5%, so we'll go below that and look at the rare SNPs, and we will also look at single measure ABPM and other finer blood pressure phenotypes that have higher heritability, as you can see here. So in summary, it's very clear today that there is no blood pressure gene. There are multiple common genetic variants for blood pressure. The effect sizes of all risk alleles seen up to now in the general population are very small, and a genetic risk score of 29 SNPs is associated with target organ damage, but not with chronic kidney disease or any other measure of kidney function. And important for you who work in the lab, or you can tell your colleagues who do, all these results are publicly available on our website in Geneva, and full results can be obtained from a publicly managed database. I'm particularly indebted and grateful to my division chief, who permits me to let all this happen. It takes a lot of time, and there is a lot of support, which is great, particularly in our institution. University of Geneva has been of great help. We receive funding of many sources, Swiss National Foundation before all, but also Geneva University and others. And there are many collaborators. Our paper had 350 authors, but this is how it should be, because many people contribute and need recognition for their contribution. Thank you very much.
Thank you, Georg, for keeping the time. As usual, as a rule, there is no questions to the speakers. And it's my other pleasure to now ask Dr. Fabrizio Montecucco, who is an MD and a PhD in our cardiology research, biology,